So let me begin by saying that I have no financial disclosures. On the other hand, I do have to admit that I have had this long-standing relationship with UCLA and with the Department of Pediatrics. My dad was the chair of the Department of Pediatrics at Harvard UCLA for 10 years, and then was the executive chair, held the position that Shirin holds now, for 9 years up until 1976. And I've had a number of other family who have been students or staff affiliated with UCLA over the years. So I have enormous affection for this institution and a real sense of loyalty. Let me set the stage for my presentation this morning by presenting this case. JD was a 23-month-old, previously healthy boy who presented to the emergency department with a 4-day history of limp and low-grade fever. In addition, he had a 1-day history of swelling of his right knee and refusal to bear weight. His immunizations were up to date. He attended daycare 3 days per week. He'd had URI symptoms for about a week, resolving 3 days prior to this presentation. And he had 1 loose stool 2 days prior to this presentation. On examination, he was febrile with a temperature of 38.7 degrees, a heart rate of 130, a normal respiratory rate, and normal blood pressure. He was alert and irritable, but consolable. He had no skin rash, normal tympanic membranes, supple neck, normal cardiac exam, normal abdominal exam, and an intact neurologic exam. On exam of his extremities, it was apparent that he was refusing to stand. He had moderate swelling of his right knee with a ballotable patella. He had warmth of that knee, reduced range of motion, no erythema, no joint line tenderness. Given his fever and joint swelling, most prominent on the differential diagnosis was septic arthritis, but other considerations included Lyme arthritis, potentially reactive arthritis, in particular considering this loose stool, possibly osteomyelitis, and probably less likely fracture, hemarthrosis, or juvenile idiopathic arthritis. With this differential diagnosis in mind, he had a laboratory evaluation that included a CBC with a modestly elevated white count of 14.5 and some left shift with 68% polys and 5% bands. He had a normal hemoglobin and a normal platelet count. His sedimentation rate was moderately elevated at 40, and his CRP was likewise elevated at 2.5. He had an x-ray of his right lower extremity given the swelling of this knee, which revealed no fracture, no bony lesions, and a moderate right knee effusion. And given the presence of that effusion and the concern about septic arthritis, he underwent an arthrocentesis, which revealed purulent fluid with 110,000 white blood cells, 92% of which were polys. He had 100 red blood cells. Gram stain of that joint fluid revealed white blood cells but no organisms. Given the concern for septic arthritis, he was started empirically on antibiotic therapy with vancomycin and cefotaxime. His joint fluid was cultured, and that culture was positive, yielding small gray colonies with a weak zone of hemolysis, as highlighted in this slide. Gram stain of these colonies revealed gram-negative short bacilli, with many in what might be referred to as a boxcar configuration. Ultimately, these organisms were identified as Kingella Kingae, and he was diagnosed with Kingella Kingae septic arthritis. So with this background, my objectives this morning will be to begin with a review of the changing microbiology of bone and joint infections in children. Next, I'd like to focus on Kingella Kingae as an emerging cause of disease in pediatric patients. And then finally, I'd like to share some insights into the pathogenic determinants of Kingella Kingae disease. Now, I want everybody to shift back to the 1980s when Shirin was in the middle of her training to think about osteomyelitis and septic arthritis. This slide summarizes the results of 3 different studies examining culture-confirmed osteomyelitis in children, all 3 of which were performed before 1990, highlighting the distribution of organisms associated with osteomyelitis. And as you can see, in all of these series, Staph aureus was the predominant pathogen, accounting for anywhere between 67% and 89% of culture-confirmed cases. In addition, Haemophilus influenzae, in particular Haemophilus influenzae type B, was a relatively common cause of osteomyelitis, accounting for between 4 and 8% of cases. What about septic arthritis? This is a slide that summarizes a study performed by Leslie Barton and colleagues and is really representative of similar studies during this period of time. This study examined culture-confirmed septic arthritis among children less than 2 years of age, between 2 and 5 years of age, and older than 5 years, and examined the distribution of pathogens during the period between 1973 and 1985. You can appreciate that Haemophilus was the most common cause of septic arthritis during this period among children younger than 2 years of age and between 2 and 5 years old. Staph aureus was second most common, accounting for about a third of culture-confirmed cases in these age ranges, and was far and away the most common cause of septic arthritis among children older than 5 years, accounting for over 90% of cases. Now, you all know that we've seen a marked reduction in Haemophilus influenzae type B disease, really beginning in the late 1980s and continuing through the 1990s, reflecting the introduction of conjugate vaccines against Haemophilus influenzae type B. You'll recall that conjugate vaccines were first licensed against Haemophilus in 1987 and at the time approved for use in children 18 months of age and older. 2 years later, in 1989, they were approved for use in children 15 months of age and older. And finally, in 1990, they were approved for routine use among infants. And it's since that time that we've seen a virtual disappearance of invasive Haemophilus influenzae type B disease, including Haemophilus influenzae type B, septic arthritis, and osteomyelitis. And I suspect that there are a number of residents in the room who have never seen a case of invasive Haemophilus influenzae type B disease. Now, beyond the virtual disappearance of invasive Haemophilus influenzae type B disease, in the late 1990s, we began to see an emergence of methicillin-resistant Staph aureus disease, and in particular, MRSA disease that appeared to be community-acquired. You may recall that the first report in Pediatrics of community-associated or community-acquired MRSA was published in JAMA in 1998 and was a paper that was first authored by Betsy Harold. Shortly following that paper, there were reports of community-associated MRSA among patients, pediatric patients throughout the United States and all over the world. And in a number of centers, we now know that community-acquired MRSA is responsible for up to 75% of Staph aureus bone and joint infections in children, with examples including reports from Houston, from Memphis, and Italy. So in this era of post-Haemophilus influenzae type B disease, of community-acquired MRSA disease, in the early 2000s, Verdier and colleagues at Dubrows Pediatric Hospital examined what's the current microbiology of bone and joint infections in children. This group carried out a study over a 3-year period between January of 2001 and February of 2004 involving 171 cases of clinically diagnosed osteomyelitis or septic arthritis, with these patients ranging in age between 9 and 90 months. They obtained fluid samples from joints, from bones, and cultured these samples, and then they subjected all culture negative samples to PCR with broad-range 16S ribosomal RNA primers. Using this approach, by culture, they were able to identify 30 cases of Staph aureus, 10 cases of group A streptococcal disease, 9 cases of Kingella Kingae disease. In total, 64 of the 171 cases were culture positive. So about 40 to 45% of all cases were culture positive. When they added their PCR results, they were able to detect another 15 cases. Interestingly, All 15 of these PCR-positive, culture-negative cases were Kingella Kingae, bringing the total of microbiologically confirmed cases to 79 and making Kingella responsible for about a third of those 79 cases. Subsequently, Shomaton and colleagues conducted a follow-up study at the same hospital, this time involving a 2-year period between January of 2004 and December of 2005. Over this 2-year period, they identified 131 cases of bone or joint infection. They again subjected these samples from bones and joints to culture, and then had all and then performed PCR on all culture-negative samples using initially 16S ribosomal RNA PCR and then Kingella Kingae specific real-time PCR. With this approach, they were able to identify a pathogen by culture alone in 45% of cases, including 25 cases of Staph aureus, 20 cases of Kingella Kingae, 7 of Group A Streptococcus, 3 of Strep pneumoniae, 3 of Group B Streptococcus. Broad-range 16S ribosomal RNA PCR was positive in another 16% of samples, in all cases Kingella Kingae. And then Kingella Kingae real-time PCR was positive in another 5% of samples. So in total, there were 87 out of the 131 cases that were either culture or PCR positive. And what they found was that Kingella Kingae accounted for the largest percentage, roughly 40% or so of those positive samples, with Staph aureus second in line. Looking at this study and the age-related incidence of disease due to a specific pathogen, they found that Kingella Kingae, shown in the black bars here, accounted for the vast majority of cases between 6 months and 3 years of age, about 80% of cases in this age group. After 3 years of age, Staph aureus, shown in the stipple bars, became the predominant pathogen. So with this information in mind, what do we know about Kingella Kingae? Well, this is an organism that was first isolated in 1960 by Elizabeth King at the CDC. Based on phenotypic properties in the clinical microbiology laboratory, it was originally assigned to the genus Moraxella, and it was named Moraxella Kingae in recognition of Elizabeth King. Subsequently, based on additional phenotypic properties, biochemical analysis, DNA studies, it was transferred to a separate genus, now in the family Neisseria in 1976, and it was renamed at that time Kingella Kingae. Since the early 1990s, there have been increasing reports of Kingella Kingae infection in the Middle East, in Europe, in South America, in Australia, and there are emerging reports in this country as well, as people have begun to use PCR. So in thinking about the specific diseases that Kingella causes, we've talked about already septic arthritis and osteomyelitis. In addition, this organism is a prominent cause of intervertebral disc infection, referred to as discitis. It's also an important etiology of occult bacteremia. It's an occasional etiology of endocarditis in both children and adults, and it's been reported on occasion as a cause of meningitis, of ocular infections, and pneumonia. In looking more specifically at Kingella septic arthritis and osteomyelitis, it causes roughly 2 to 3 times more cases of septic arthritis than osteomyelitis. Septic arthritis most often involves the bones of the lower extremities, most commonly the knee, next most commonly the hip, and then the ankle. Kingella osteomyelitis most commonly involves the long bones of the lower extremity, specifically the femur and the tibia. And interestingly, this organism also has a predilection for producing infection in the small bones of the foot, specifically the calcaneus and the talus. This slide highlights that, again, the disease is most common in children in the first 3 to 4 years of life. The peak incidence of disease is in the age range between 6 months and 17 months of age. It's very uncommon during the first 6 months of life, and it's very uncommon after 3, 3.5, or 4 years of age. At least a partial explanation for this age-related incidence is the fact that there is antibody that is present against whole Kingella organisms during the first 6 months of life, likely reflecting transplacentally acquired antibody. And then there is the emergence of antibody by about 2 years of age. Whether that's related to exposure to Kingella because of colonization with Kingella, or instead exposure to cross-reactive antigens that provide some level of protection, unclear at this point. Now, like other important causes of invasive disease in pediatric patients, with examples including Haemophilus influenzae type B, Neisseria meningitis, Kingella appears to be capable of efficient person-to-person transmission and capable of producing outbreaks of disease. And the first reported outbreak of invasive Kingella disease was described by King et al. From Minnesota, published in Pediatrics in 2005. This outbreak involved 3 cases of Kingella osteomyelitis and septic arthritis among children attending the same daycare center. Studies of the other 20 children in this daycare center revealed that 9 of the 20 were positive in pharyngeal cultures. And all of the isolates recovered here, including the invasive isolates as well as the pharyngeal isolates, appeared identical by pulse field gel electrophoresis, suggesting that they were the same strain that was being transmitted from patient to patient in this group. The second report of an outbreak of Kingella Kingae invasive disease was described by Yagubski a year later, involving 3 children in Israel. These 3 cases were all osteomyelitis and occurred over a 15-day period involving children in the same daycare center. 4 of the other 11 children in this daycare center had positive pharyngeal cultures. And again, all isolates, both the invasive isolates associated with disease and the pharyngeal isolates associated with colonization, were identical by both pulse field gel electrophoresis and random amplified polymorphic DNA analysis. Since this time, there have been another at least 8 reports of outbreaks of invasive Kingella Kingae disease. I've mentioned that this is an organism that appears difficult to cultivate, presumably because it's a fastidious organism like other Neisseriaceae. And with this information in mind, some years ago, Yagubski and colleagues wondered whether there might be a way to enhance recovery from the organism, perhaps by inoculating joint fluid samples into blood culture bottles. So specifically, in this study, Yagubski and coworkers obtained joint fluid and then simultaneously cultured joint fluid from a given patient on solid media using traditional approaches and inoculated some of that joint fluid into Bactec blood culture bottles. There were a total of 11 patients in this study who had positive cultures. 1 of these 11 had a positive culture both on solid media and in a Bactec blood culture bottle. And the other 10 of 11 had negative cultures on solid media and positive cultures only in the Bactec blood cultures, in the Bactec blood culture bottles, suggesting that blood culture bottles may be a way to improve detection, improve recovery. Since that time, others have looked at a variety of other blood culture systems, and there are a variety of other systems that also appear to increase the yield of cultivation, including the isolator culture, as well as the BacT-Alert, the BacT-Alert fan, and potentially the BacT plus aerobic F. But despite the ability to enhance recovery using blood culture bottles, culture remains a relatively insensitive method to detect this organism. And the ability to detect Kingella is markedly improved by using PCR, as highlighted in this slide, which shows results from 12 different studies published over the last 8 or 9 years or so. In total, there were, in these 12 studies, 223 patients who were diagnosed by either culture or PCR or both with Kingella Kingae infection. Roughly 18% of these positive samples were positive by culture, and all but 1 were positive by PCR, emphasizing that PCR is a much more sensitive way to detect this organism, at least in skeletal infections. Well, what about the approach to treatment of Kingella? Most isolates are highly susceptible to penicillin, to ampicillin, to first generation, second generation, third generation cephalosporins, to trimethoprim sulfamethoxazole, to ciprofloxacin. There have been increasing reports of beta-lactamase production with activity against penicillin, ampicillin, cefazolin, but most of these reports describe low-level resistance, not clear that it's clinically significant. There also are reports of resistance to oxacillin and trimethoprim sulfamethoxazole and ciprofloxacin. Importantly, as we think about the approach to treatment of community-acquired MRSA, it's important to recognize that most isolates, or at least many isolates, ranging between 40% and 100% of isolates, depending on the series, are resistant to clindamycin, a common drug to choose for treatment of MRSA infection. And Kingella isolates are uniformly resistant to vancomycin, again, a drug that we think about commonly in the setting of Staph aureus infection when we're worried in particular about MRSA infection. Well, in thinking about the pathogenesis of disease due to Kingella, Yagubski speculated that perhaps like, again, a number of other invasive pathogens in pediatrics and pediatric patients, Kingella may initiate infection by colonizing the pharynx. And in order to address this hypothesis, he and colleagues conducted a study that involved collection of pharyngeal and nasopharyngeal samples every 2 weeks over an 11-month period involving 45 children between 6 and 42 months of age, all attending the same daycare center. During the course of this study, these investigators found that 27.5% of pharyngeal cultures, but no nasopharyngeal cultures, grew Kingella. The monthly prevalence of Kingella colonization ranged between 6% and nearly 35%. And there was colonization at some point during this 11-month period in over 70% of these children. So colonization of the pharynx appears to be quite common, at least in the population that was examined by Yagubski and colleagues in this study. Perhaps a stronger link between pharyngeal colonization and invasive disease comes from this report of 3 children who had simultaneous blood cultures and pharyngeal cultures. These 3 children included a 12-month-old with tenosynovitis, a 10-month-old with bacteremia, and a 6-month-old with bacteremia. And all 3 of these patients had blood cultures and simultaneously a culture from the pharynx. And in all 3 cases, the isolates from the systemic site and the pharyngeal site were identical based on pulsed field gel electrophoresis and random amplified polymorphic DNA, making the suggestion or supporting the suggestion that disease begins with colonization of the pharynx and subsequently involves invasion of the bloodstream. So with this background, I'd like to shift at this point and spend the next 15 or 20 minutes or so talking about work in my laboratory that has begun to explore how this organism produces disease. And I'll highlight work by 3 different individuals who have been graduate students in my lab at some point during the last 10 years or so, including Thomas Kielfai, who initiated the work that we've been doing on Kingella and now is a member of the faculty in the Department of Microbiology and Immunology at the University of Illinois. Eric Porsche, who really took the baton from Thomas and now is a member of the staff still in my laboratory at CHOP. And then most recently, Kim Starr, who just defended her thesis at Duke and now has a position in the clinical microbiology lab at Duke. So we initiated studies by examining the ability of Kingella to adhere to human respiratory epithelial cells, aiming to identify the determinants of Kingella colonization of the pharynx. And in these studies, we took a clinical isolate of Kingella, referred to as KK269492, originally isolated from joint fluid of a child with septic arthritis involving the knee, and then inoculated this organism, this strain, onto a monolayer of cultured human respiratory epithelial cells. And as you can see on the right-hand side of this set of light micrographs, Kingella, this strain of Kingella, was capable of very efficient adherence to these cells. As a control in this experiment, we used a laboratory strain of E. coli, and you can appreciate that E. coli was unable to adhere to these cells. In thinking about the mechanism by which Kingella might be able to adhere to human respiratory epithelial cells, colonize the pharynx, we wondered about the possibility that this organism expresses adhesive pili. And there had been early reports that indeed Kingella, or at least some isolates of Kingella, do express these thin hair-like structures called pili with adhesive activity. Along these lines, we examined our prototypic isolate by transmission electron microscopy after staining with uranyl acetate. And indeed, we were able to demonstrate that this organism does express pili. And the morphology of these pili has a resemblance to a class of pili referred to as type 4 pili. In fact, a variety of Neisseria species express type 4 pili, including Neisseria gonorrhoeae, Neisseria meningitis, and we've learned a fair bit about the expression of these polymeric macromolecular structures in Neisseria. In Neisseria, type 4 pili are comprised of a major structural subunit called pil-A, and the assembly process involves a cytoplasmic ATPase called pil-F, an intermembrane peptidase called pil-D, and an outer membrane pore-forming protein called pil-Q. To facilitate our studies of Kingella, we sequenced the genome of this clinical isolate, and then we examined this genome for potential homologs of known type 4 pilus-related genes. And indeed, we found that there were homologs of pil-A1, of pil-F, of pil-Q, pil-D. With this information in mind, we insertionally inactivated the Kingella pil-A1 gene and the Kingella pil-F intermembrane ATPase. We then examined these mutants by transmission electron microscopy and confirmed, as expected, that both were non-pileated. And then we examined these non-pileated mutants, the pil-A1 mutant, the pil-F mutant, in adherence assays, as shown in this plot of adherence with adherence as a percent of inoculum shown on the y-axis. And you can appreciate here that in assays with 3 different cell lines, Chang, A549, and Hep2 cells, the non-pileated mutants had lost the ability to adhere to these cells. In support of the possibility that indeed pili are important in the process of colonization of the pharynx, we examined 28 pharyngeal isolates by transmission electron microscopy, a relatively insensitive way to appreciate the presence of pili for pili, and we found that 80% of these isolates had visible pili. As a next step, we wondered whether there might be other factors that also influence adherence to respiratory epithelial cells, colonization of the pharynx. And in an effort to identify other factors, we began by generating a transposon library using a transposon system referred to as the Mariner system. We generated a library of mutants, and then we screened these mutants for examples that were non-adherent. And using this approach, we identified a number of mutants involving insertion of the Mariner transposon element into a number of different genes. We were especially intrigued regarding the mutant with an insertion in the gene that encodes a so-called NhhA-like protein. Now, NhhA is an adhesin that's expressed by Neisseria meningitis and has been demonstrated to play an important role in meningococcal adherence to epithelial cells. It is also a potential vaccine antigen for protection against meningococcal disease. And as highlighted in this slide of 2 crystal structures, the NhhA protein is a member of a trimeric family of proteins called trimeric autotransporters. When we insertionally inactivated the NhhA-like protein in Kingella, which we designated KNH for Kingella NhhA homolog, we found that there was a marked decrease in adherence. In this slide, we again show adherence on the y-axis as a percent of inoculum, adherence to 2 different cell types, Chang cells and A549 cells. We see adherence by the parent strain on the left, and then adherence by the KNH mutant in the middle, with a marked reduction, 80 to 90% relative to the parent strain. So based on these studies of type 4 pili and of KNH, it appears that both type 4 pili and KNH are required for full-level adherence to human respiratory epithelial cells. But in considering these results, two questions came to mind. First, why does our non-pileated mutant lack adherence if KNH is present, and indeed KNH has adhesive activity? And as a corollary, is there another surface factor that might block KNH-mediated adherence when pili are absent? Well, in pondering these questions, Thomas, at one point, inadvertently left plates of Kingella on his bench for a longer period of time than usual. And when he looked at those plates, he noticed that the Kingella colonies were much more mucoid than he had appreciated before. And this mucoid morphology suggested the possibility that Kingella expresses a polysaccharide capsule. It had not been known to be encapsulated prior to this work. But with this idea in mind, we began by staining our prototypic strain with cationic ferritin. Cationic ferritin is a positively charged stain that has an affinity for negatively charged polysaccharide capsules or polysaccharides. We then thin section these samples and examine them by transmission electron microscopy, and we appreciated this electron-dense halo around the organism, exactly as one typically sees with an encapsulated bacterial pathogen. To extend this work, we looked at the Kingella genome that we had and looked specifically for genes that are homologs of genes known to be involved in capsule expression in other bacteria. And interestingly, we found a cluster of genes referred to as the CTR-ABCD genes that have homologs, highly homologous homologs in Neisseria meningitis involved in the export of the Neisseria meningitis capsule. So we insertionally inactivated the CTRA gene, and then we examined whether disruption of this gene eliminates capsule, or at least eliminates the mucoid colony morphology and eliminates this electron-dense halo around the organism as appreciated after cationic ferritin staining. And here on the left side of this slide, you can appreciate that elimination of CTRA indeed changed that colony morphology and eliminated the mucoid appearance. And similarly, insertional inactivation of CTRA eliminated the electron-dense halo after cationic ferritin, providing very strong evidence that indeed Kingella is encapsulated. So, what's the influence of capsule on KNH-mediated adherence? And in particular, how do we explain the fact that the non-pileated strain that still expresses KNH is non-adherent? Well, you can see here that when we insertionally inactivated pili-1 and eliminated pili, and also eliminated capsule, we were able to restore the adherence in the absence of pili and the presence of KNH. And as evidence that indeed this adherence is KNH-mediated, when we also insertionally inactivated KNH, we completely abrogated adherence. So how is it that capsule interferes with KNH-mediated adherence in the absence of pili? Well, we wondered about the possibility that Kingella pili might be able to undergo a process referred to as pilus retraction, a phenomenon that's been well described in type 4 pili in a number of other organisms. And in other organisms that express type 4 pili, this process of retraction or withdrawing pili into the organism is mediated by an intermembrane associated ATPase called PIL-T. And in fact, Kingella possesses a PIL-T homolog. So we insertionally inactivated that PIL-T homolog, and as shown in this plot of adherence, that PIL-T mutant had a level of adherence really almost exactly the same as elimination of KNH, consistent with our hypothesis. So with this information in mind, we proposed the following model for how Kingella interacts with respiratory epithelial cells and potentially how Kingella colonizes the pharynx. we suggest that initially Kingella makes contact with the host cell surface via type 4 pili. Subsequently, these type 4 pili undergo retraction, drawing the organism closer to the host cell surface, displacing the polysaccharide capsule, allowing exposure of KNH and allowing subsequently KNH to engage with the host cell receptor. And we're currently in the process of pursuing some other studies to indeed confirm this model. In fact, we think this model probably applies to a number of other bacterial pathogens as well. In particular, when we think about pathogens that express pili, that express a non-pilus adhesin, and that express a capsule. And examples of organisms that satisfy these criteria include Neisseria meningitidis, enteropathogenic E. coli, an important cause of infantile diarrhea, in particular in developing countries, Vibrio cholera, a cause of cholera, and Moraxella bovis, an important dog pathogen. So if we think about the pathogenesis of disease, we've laid out a series of factors that may participate in the process of colonization. Subsequently, the organism needs to translocate across the epithelial barrier in order to enter the bloodstream and produce systemic disease. And considering that step, it's noteworthy that in all the studies that I've shared so far, we chemically fixed monolayers before inoculating bacteria and examining adherence properties. But in fact, when we omit chemical fixation and then inoculate Kingella onto a monolayer of viable epithelial cells, we appreciate a very potent cytotoxic effect, as shown in these light micrographs. We see a monolayer after inoculation with a laboratory strain of E. coli on the left that's perfectly intact, and we see a monolayer 5 minutes after inoculation of Kingella on the right. And you can appreciate that there's been rounding of cells and lifting of many of those cells off the plastic support. So in fact, this cytotoxic effect is correlated with cellular release of lactic dehydrogenase, and we can detect that lactic dehydrogenase using a spectrophotometric assay, as highlighted in this slide, which shows results with 5 different strains and 5 different Kingella strains and 3 different cell lines, demonstrating that all of these strains with all of these cell lines are cytotoxic. So in considering the mechanism by which Kingella mediates the cytotoxicity and potentially is able to translocate across the epithelial barrier, we again took advantage of our Transposon mutant library, this time screening for mutants unable to induce cell lysis and release of lactic dehydrogenase. And when we did this screen, we identified 3 mutants designated 60H11T1, 69A6T1, and 97G1T1 with reduced cytotoxic activity, with reduced ability to lyse cells and release LDH. When we mapped these the insertions in these 3 mutants, we found that all 3 insertions localized to the same region of the chromosome and inserted into genes that are associated with expression or have homology to genes involved in the expression of a family of cytotoxins referred to as RTX toxins. RTX toxins are expressed by a variety of different gram-negative pathogens, have a series of calcium binding repeats, and bind calcium, undergo conformational change, insert into membranes, and produce a pore in host cell membranes, resulting in this cytotoxic effect. Now, interestingly, the RTX genes in Kingella are most homologous to genes in Neisseria meningitidis and this dog pathogen, Moraxella bovis. Here we show gene products and the percent identity, percent similarity. You can see that the percent identity ranges between 64% and 83%. The percent similarity ranges between 82% and 91% among these Kingella genes and the homologous genes in Neisseria and Moraxella. We next wondered what's the prevalence of RTX genes in a range of Kingella strains. And we performed southern hybridization studies to address this question, probing with an RTX-A, RTX-C fragment of DNA. And as highlighted in this slide, we found that all Kingella Kingae strains have these RTX genes, but none of the less pathogenic Kingella species harbor these genes. The suggestion from this experiment is that an ancestral strain of Kingella at some point along the way acquired the genes required for expression of an RTX toxin from perhaps Meningococcus, perhaps a related Moraxella species, and created Kingella Kingae now with enhanced pathogenic properties. So in thinking further about the pathogenesis of Kingella disease, once the organism enters the bloodstream, it needs to survive, to replicate, and then to disseminate to distant sites in order to produce disease. And in thinking about the mechanism by which it survives, replicates, and disseminates, we wondered again about the role of the capsule. This is a slide that summarizes results in an infant rat model of invasive disease involving intraperitoneal inoculation of the organism. And you can appreciate here that the wild-type organism produces fatality in almost all animals in 24 to 30 hours or so. In contrast, when we insertionally inactivate CTRA and eliminate encapsulation, the organism seems non-pathogenic and has a phenotype analogous to inoculation of just buffer alone. So in order to gain a greater knowledge of the Kingella capsule, we purified the capsule from our prototypic strain, KK269492, and then performed composition analysis and structural analysis of this capsular material. And we were able to establish that this capsule is a polymer of N-acetylgalactosamine and a molecule called 3-deoxy-D-mannooxyloxonic acid, which is abbreviated KDO and is an important component of LPS in many organisms. In thinking about what other factors might be involved in generating a capsule, you'll recall we've talked about the CTRA-ABCD genes, and closer examination of the Kingella genome revealed genes called LipA and LipB that have homologs in Neisseria meningitidis and E. coli that are involved in the capsule assembly process. And using our Transposon mutant library, we were able to identify another gene that we designated CSA-A for capsule synthesis. This gene encodes a protein that appears to be a glycosyltransferase and appears to have two functional domains. There's an N-terminal domain that has homology with galactose transferases, and we think this domain likely mediates transferase of the N-acetylgalactosamine moiety to the membrane poly-KDO linker. There's a middle domain that has homology to KDO transferases, and we think this domain is responsible for linking KDO to N-acetylgalactosamine. Subsequently, the galactose transferase again mediates linkage of another N-acetylgalactosamine to KDO, and so on and so forth. Well, what about the diversity of Kingella Kingae types? Do all organisms express a capsule? Is it the same capsule in all organisms? So to address this question, we generated a series of PCR primers, including a set of internal primers internal to this, the CSA-A gene, and external primers that are based on flanking genes. And then we screened a large collection of clinical isolates. And what we found was 3 different genetic configurations that ultimately we designated type A for our prototypic strain, and then types B, C, and D. When we did composition analysis, structural analysis on the types B, C, and D capsules, we found that the type B capsule is a polymer of N-acetylglucosamine and KDO. The type C capsule is a polymer of ribose and KDO with 3 ribose moieties in the repeating unit. And the type D capsule is a polymer of galactose and N-acetylglucosamine and is phosphorylated. With this information in mind then, we screened a large collection of carrier isolates, a total of 246 isolates recovered from the pharynx of individuals who are colonized, and then 181 isolates from patients with invasive disease. We showed the type A prevalence with a dark gray bar, the type B with a light gray bar, the type C with a white bar, and the type D with a black bar. And you can appreciate that among carrier isolates, there's a nice distribution of all 4 of these capsule types. In contrast, among invasive isolates, we see that almost all isolates express either the type A or the type B capsule. Over 95% of cases express the type A or the type B capsule. The suggestion here is that potentially the type A, type B capsules are associated with enhanced virulence because of specific properties of those capsules, or perhaps those capsules are linked to other genes present in those specific clones. Alternatively, it may be that the type C capsule and the type D capsule are associated with properties that allow the organism to survive more effectively in the posterior pharynx. So let's just review what we understand then about the pathogenesis of Kingella Kingae disease and the various Kingella factors that are involved in each of these steps. So as shown in this schematic, the process of disease begins with colonization of the posterior pharynx. And we have evidence based on our in vitro studies that type 4 pili play an important role in this process, that KNH plays an important role in this process, and that the capsule interferes with KNH-mediated adherence, in particular in the absence of pili. Subsequently, the organism invades the bloodstream, translocates across the epithelial barrier within the posterior pharynx, and we propose that the RTX toxin plays an important role in that process. A variety of epidemiologic studies have demonstrated that most patients with Kingella Kingae invasive disease have a concomitant respiratory virus or oral viral infection. And it may be that there's a special dynamic that takes place between the RTX toxin and either the viral infection or the consequence of the viral infection that allows invasion of the bloodstream. And then finally, we present evidence that the capsule plays an important role in allowing the organism to survive inside the bloodstream and replicate and then disseminate to distant sites to produce septic arthritis, osteomyelitis, endocarditis. So let me conclude by summarizing what I've shared this morning. I've described the microbiology of osteoarticular infections and highlight that it has changed significantly over the past 15 years. The key factors influencing this change include routine immunization against Haemophilus type B, which has largely eliminated Haemophilus influenzae type B as an etiology, the emergence of community-acquired MRSA, and the development of improved diagnostic methods for Kingella Kingae. In addition, it appears that important determinants of Kingella Kingae pathogenesis include type 4 pili, the KNH adhesion, a polysaccharide capsule, and an RTX toxin. So where will we go from here? Well, we're very interested in thinking about Kingella as a model pathogen, an organism that is largely a commensal organism, but under the proper circumstances is able to transition from commensalism to disease. And we know that this is the case for the most common invasive pediatric pathogens in our patient populations, including Neisseria meningitis, Streptococcus pneumoniae, Staphylococcus aureus, Group A Streptococcus, Group B Streptococcus as examples. We're also interested in understanding the mechanism by which Kingella induces inflammation, and we think that the RTX toxin is likely to play a role in that process. And then finally, we're interested in understanding more about the immunogenicity of the Kingella polysaccharide capsules, thinking about the possibility that Kingella is as prevalent in this country as it is in other countries, and that there may be some rationale over time to develop a vaccine against this organism. So on that note, let me thank you for your attention this morning. I'd be happy to answer any questions.